Well, I think I can just thank the organizers for inviting me to share with you before the mic is on. And then moving on. Oh, sorry. Excellent. Sure. So I'm aware we're behind schedule, so I'll try to do a quick whistle stop in what we're trying to do with open science and bringing you targets for therapeutics. So today we're talking about, of course, precision medicine. And I think this picture illustrates really well what people understand about precision medicine, which is actually identifying the best group that will benefit from different targeted therapies. However, when we look at that, what we see is just a subset. In a recent study by MD Anderson, they looked at about 2,000 patients and actually using a standardized hotspot mutation analysis, looking at 46, 50 genes with the known mutation that would cause cancer, they were only able to map 6.4% of all these patients to a target drug. That means that, as a consequence, all these are left untreated. It's not a gloomy image. For the patient itself, for the individual patient, it's good news that we have a drug, but we cannot leave these guys behind. And I would like just to conclude today's discussion on precision medicine on what the SGC is trying to do to help these populations here. So despite all the challenges, innovation about medicine, oh my goodness, is really risk averse. Different talks today actually mention different kinases. And if you look at all the human kinases in the human genome, there are about 520 of them, and map against all the papers published on these 520 kinases, you see that 90% of all the different papers and publication studies made were in less than 10% of those kinases, of these potential targets. Even though you have lots of evidence now that show that many of the other kinases are also important. If this is only in academia, it's fine. We would be hoping that the industry will be trying to tackle all these novel targets. However, when you look at the patents found around those same kinases, well, it follows the same trend. So we're in a stage where there's a very little appetite for high-risk targets. We cannot convince from the agencies or research directors in pharma to sponsor studies here, because where's the evidence, where's the return on investment? And to make things worse, we work in a very over-competitive mode. We share very little with other colleagues to tackle very difficult problems. So one of the solutions that could be brought to the table, and one thing that many groups are advocating for it, is actually creating public-private partnerships where resources are pooled to actually tackle try to work only on novel proteins, those proteins where very little body work's been done, and generating toolkits and basic knowledge informing the human protein structures, chemistry around those targets to modulate their function, antibodies, screening, cell assays, and so on, and more importantly, make them public available, i.e. no patents, no restraints on use. Just see it out there instead of actually trying to hold it back. Why? Because we don't understand enough human biology. It's very clear that one of the reasons we do precision medicine now is to really figure out what is the subpopulation that drug will be working on, which is a pathway that drug is touching on. That only means that we don't know enough biology. So why not share this with the entire public domain to finally identify a proof of concept and then move to the proprietary route. Now, this is actually what the SGC has been doing since 2004. We are a public-private partnership. We are based in several universities, and the largest site here in Oxford, 140 scientists, funded by government agencies. That includes Canadian ones, one in Brazil, Wellcome Trust here, the IMI. And together, with all 6 labs, we do exactly this. Open access research on understanding targets, and actually promptly placing all the results, reagents, and know-how in the public domain. And we will never file for patents, because we believe that that's just waste of time to actually spend a lot of energy in this such an early stage. And you might be interested to see that there are quite a few pharma partners funding this effort, even though we clearly said there's no patents around. Does that work? It's not just a model. So in order to show you, I would like to share with you a case study where this happened. So many people here have been talking about genetics. We know there's not a layer of information, the epigenetics. When we talk about epigenetics drugs, back in 2009, 2008, most of the research revolved around HDAC inhibitors, which was the only class of epigenetics regulators that were actually being targeted. Even though, and that covers about 25, 30 genes, proteins. Even though the entire repertoire of proteins involved in epigenetics regulation is actually up to 400. So we decided to work on a new class called broad domains. These are readers of the epigenetics regulation of histones. And as we can do structural biology, solving the 3D structure of those targets very quickly, we went to the human genome list, 61 targets. We solved 35 of them. And you can see how they look like and start designing compounds for it. So the program started about 2009. We designed a compound and actually started looking for a group that actually had a case, clinical case of one of these targets, in this case BRD4, which actually appeared in a fusion, as a fusion protein, in a rare cancer called nut midline carcinoma. It all happens that Jay Bradner in Dana-Farber actually was working on it. So we contacted Jay and said, oh, why don't we start collaborating? We have the compound, you guys have disease models, you have the cancer, rare cancer, you have the xenograft models. Should we test it? He said, yes. Should we get the noising? No, we don't need to, because there's no patent, right? Here we go, hang up the phone, send the compounds, and between that and doing all the studies, write the paper, send it to Nature, published in 11 months. That's the acceleration of the process. And look and behold, the compound, we showed everybody what it looks like. We're not holding back. Compound when dosed actually reduced the tumor, increased the apoptosis, and increased the differentiation. Also, what happens is that GSK working with us started testing this also in the non-disease model information. Not only we show what the structure looked like, we also said, if you want a piece of the action, write us. We'll give a sample of the compound. And then we start sending compounds after a month to more than 100 labs. What these labs are working on, we don't know. They are probably the best experts in different disease areas. But now they have a compound which was designed specifically for one target. That nobody knew what's the biological function. They started testing on different disease models and start seeing effects. Very quickly, it lasted 6 months, new papers came out. Who would have thought that BRD4 is actually linked to leukemia, is linked to myeloma? For the first time, we're now looking at different pathways that are actually linked to these disease conditions. Of course, one of our pharma partners said, well, it's a completely new target, it's a completely new molecule, it could be an off-target effect. Let me do it, help you guys. We designed another compound for the same target, and under the SPC principle, also gave it away. No patent. Go, do the test. Compare the data. Validate it. So what happened is there's so many people now getting access to this new compound, this new biology, very easy to publish in Nature Cell and Science. Everybody started publishing. So much so that GSK looked at it, said, that's enough data. We believe in this. Let's go to clinical trials. First in man, first in class. Less than 3 years after we started the project. All right? Now, compare this to typical 6 to 30 years for many of the kinase projects. That's a significant reduction of the acceleration of completely new target class. What happens after that? The paper came out in December 2010. Today, there are more than 40 clinical trials registered by many different companies with their own chemistry for that target class. This has actually already moved, if you like, more than $1 billion in investment, private investment on downstream development of this target and new compounds. Remember that graph that I showed you, the pre-competitive open space and the competitive area? That's already happening. And of course, that's the story of one probe. SGC has been doing this for the last few years, and we now put now in public domain more than 50 of those compounds for about 30, 35 different targets in the genetics regulators. And we have more to come. We really want to crack all this family. But we don't work only on epigenetics. We understand there are other diseases for which new evidence are being generated through high-throughput sequencing, through sequencing efforts. So the new genes now be linked to diseases. And what happens when you sequence and find that a gene might be related to a disease? Well, I'll tell you, a lot of this data is sitting there, waiting for some scientists to work on it. What scientists need to work on it? Reagents. Who's going to produce the reagents? Well, not me. It's very risky, very expensive. The SGC stepped up to the challenge and said, we're going to start doing this. This is a project funded by the Wellcome Trust, where we're going to start generating what we call the target enabling packages, i.e., all these reagents for completely new targets linked to disease, to genetics, that we're going to put in the public domain. Since the project started 2 years ago, June was 2 years, we've now put in public domain 15 of those target enabling packages. All the data can be seen here as well. So that actually, these are the different proteins for which we have one protein targeted package released to date. You can see these targets cover different areas: cancer, malaria, metabolic diseases, neurosciences. But the beauty of this is that different from any asset you license in from either academia or pharma, there are no constraints. Even though the entry point could be cancer, these target and this toolkit can be looked into inflammation education as well. So it's a free combination of everything that you can imagine, so the best scientists can get access to it. Also, I told you about the probes, right? Those compounds, the 50 plus compounds. Nowadays, we don't produce them. We teach Sigma, we teach Toprid, etc., how to make them. And that compound, the JQ1 that I showed you, instead of paying $1 million or $2 million for that license, you can buy for $50. So cheaper than a textbook. So any good undergrad student with good idea can actually get that and start doing it. So finally, how do we increase the power of this? We know that, well, I hope they don't look like this. Many mouse models, they don't really reproduce disease outcome, right? So instead of doing this, why not set up a very competitive primary cell platform where we can actually start looking at different compounds and link that to different diseases. So that's been funded by the IMI. It's a project we call UltraDT. And one of the final slides is actually how are we really combining the SGC platforms to increase translation. I told you about the disease platforms where we get primary cell from patients. We're going to develop new assays to try to correlate the disease. And armed with a set of probes which are specific for different targets, we can crisscross this. When we crisscross this, we start making new target disease associations. We start making new starting points for the discovery, all in the open. And finally, we have a strategic coverage of the open space. Not only we say we're going to do more exploratory work, but the entry point actually covers major therapeutic areas. All the assets generated here can be free flow and exchange among different projects. And these are different projects that are funding this effort. This is funded by the Wellcome Trust, £10 million, or £9 million. Alzheimer's Research UK with the Oxford Drug Discovery Institute for dementia targets, that's £10 million. UltraDD, which is the EU-funded project for inflammation targets, that's 50 million euros. And of course, we have a new site now in Brazil and also at UNC, where we're actually looking at those unexplored kinases. These altogether, it's about $15 million effort. With that, I would like to thank you for your attention and open the question if you have time. Otherwise, we can actually have a chat over drinks. Thank you.